Enzymes, biological catalysts. As we speak, hundreds of thousands of them are operating throughout our bodies, playing vital roles in movement, metabolism, and essentially all cellular processes. Without them, chemical reactions would happen so slowly that our cells wouldn't be able to operate, and frankly, we'd be completely stuffed. But how can we tell what effects particular enzymes are having on particular reactions? And, more importantly, why might we care? Enzymes and their action are widely studied in biochemical laboratories across the planet, and here at the University of Birmingham's School of Biosciences, it's no different. Researchers are investigating the role of a particular group of enzymes called Pyro phosphatases in establishing self-incompatibility in plants. Self-incompatibility is the mechanism by which plants are prevented from fertilizing themselves, ensuring that they only reproduce with other members of the same species. Now, this process is important for things like evolution and adaptation. In order to be able to demonstrate the role played by pyrophosphatases in self-incompatibility, it's important to be able to measure their rates of reaction. Now, this can be done using the exact same theory and equipment that we might find in an A-level biology classroom. In order to measure the rate of pyrophosphatase activity, we need some pyrophosphatase enzyme that we've isolated from plant pollen, and we also need a substrate solution that's going to change colour as we proceed through our reaction. This substrate solution contains ammonium molybdate and iron sulfate and should turn a blue colour following enzyme action. In order to detect this colour change, we're going to use a spectrophotometer, which is essentially a fancy colorimeter. To start the reaction, all I need to do is add a known volume of substrate solution to a known volume of enzyme solution in a squeaky clean cuvette, and then immediately start reading the absorbance at a particular wavelength. If I measure the absorbance at different time points, I can construct a standard curve from which I can then calculate the initial rate of reaction of my pyrophosphatase enzyme. Importantly, I want to repeat this experiment a number of times to ensure that my results are reliable. By looking at the pyrophosphatase activity under different conditions, this can help us to figure out how the protein functions in vivo, inside the plant cell, and can tell us a bit about how self-incompatibility is controlled and functions in plants. Understanding how self-incompatibility works could help researchers to disrupt this process, enabling more efficient plant breeding technologies to be developed that could help to feed Earth's ever-expanding population.